these machine members go to other places. Hi, happy Thursday. That feels like Friday because you don't have to be here tomorrow. Today, I mostly want to go over things that we've done and make sure we understand how to do those things. The first one of those things I want to do is your cell specialization worksheet. That's the one that had the 2 blanks for every item. It says cell specialization at the top. You did a Google form to get the answers, or to put your answers in. And what I discovered is essentially that I think it was a little too hard. So this is the one I'm not going to take a real grade for. But I wanted to make sure that you understood the options here and why we chose what we chose, because you could see things like this on the test. So let's have a look at these. And these are all real human cells that do exactly what I'm saying they do here. First, called the phagocyte, which is a white blood cell, sometimes called the suicide sac, which engulfs and destroys bacteria and viruses. Now, I bolded the terms that you should be kind of paying attention to. The destroys one should have been the easy one. Which organelle is a phagocyte going to need a huge one of to destroy stuff? It's like a vacuole, but the kind that kills you. Lysosome. That would be the lysosome. Yeah, and those are kind of related organelles, but the lysosome is the one that's full of nasty chemicals that can break stuff down. In fact, that's what phagocytes have. They have a big fat lysosome. Basically, they swallow enough bad guys, and then they pop their lysosome, killing themselves and the bad guys that they've swallowed. What about the engulf part? This was the tricky one, I think, where some of you might have picked cell membrane. That's not what we were after on that one. We were after the part that allows a cell to change shape enough to swallow something. Say again? It's not the mitochondria. That's the one that provides energy. What we were looking for here was the one that helps the cell change shape, which is microfilaments. Remember, those are attached to the inside of the cell and anchored to the cell membrane and can help the cell change shape when it does something like swallow stuff like we showed in the endosymbiotic theory. So it was D as in dog and F that were the correct answers for number 1. Number 2, the muscle cell. The word work, constantly doing the work of contraction should have tipped you off immediately that was what, Hayden?
That it was going to be apparatus.
Which allows a cell to do work, which provides a cell with energy, which is the powerhouse of the cell system. It is the mitochondria. Anything that does a lot of work like that is going to need mitochondria. So A is the first answer, or the second, whichever one you get. But then small fibers, well, only 2 of those count as small fibers. So what was it? What were you down to as far as choices there? 2 options. This is part of why I didn't take the grade, because it was hard for you to know which of the 2 fibers. But microfilaments or, Hermione, microtubules, you give yourself credit if you got either one of those. I didn't know how to do that in the form. Number 3, a gland cell makes hormones, which are special proteins, which are made and then moved through the cell to the surface and then sent out of the cell to cause changes in other cells. What do you think? How about making a protein? Which organelle is going to do that? Armani. Not the vacuole, doesn't make protein, right? No? Golgi apparatus is correct for the sending it out of the cell. But making proteins and then moving them through the cell, that happens on the endoplasmic reticulum. Technically, it's the ribosome that does it, That's all part of that endomembrane system. So endoplasmic reticulum and Golgi apparatus would be the most correct answers for those. That's B and C. Nerve cell, lots of energy to conduct electrical signals along its surface. A neuron is basically elongated like this. So first of all, if it's elongated like this, what does it have a ton of? What's the surface that I bolded there? Surface. That's the one you pick cell membrane for, because it's got tons of surface area. And the energy part, no brainer, right, Vinny? What is it? Mitochondria, absolutely. So A and G for number 4. And finally, number 5, you know about the sperm cell. It swims with its flagellum. Swimming is going to require a lot of what? Energy, which means, again, mitochondria. That's A. What about the flagellum? Did you take the time to look up what flagella are made of? Okay. Close.
Microtubules.
Microtubules are what flagella are made of. So A and E for number 5. Those are hard. But if you know your cell parts well enough, you should have at least done a decent job. And boy, there were a lot of zeros on this in that form. So I don't think a lot of you took the time to really connect those functions to the structures. So use that right there as kind of a study guide, because you could well see some of those items on the test, either exactly as written or tweaked a little bit. Good, next. I will tell you that the tiny bags of water assignment was much better. So that's the one we're taking a real grade on. What you'll see in Google is you should get an email when I return the scores. I'm going to do that about midday today. I'm just going to have the scores go back. I don't actually know what that looks like for you when I say return everybody's scores. Okay, so, but I saw that there were options where I could actually return it in such a way that you saw what you missed and what the correct answers were. So that is the way I will return it. So you'll see the key and be able to see what was correct and what wasn't. But again, a lot of you did great.
If you have the Google Classroom and the Gmail app on your phone, you'll get a Google Classroom, and when you Google Classroom, it tells you to go on the email.
Okay, so it takes the time to dig in and actually see what you missed, and that'll help. For now, I want you to grab your beaker problems from yesterday. Let's check those out. Okay. So on this side, is that what you've got? Yes, good. We've got 4 different solutes, and by the way, each beaker is going to be worth a half a point. I'm going to have you grade your own on this one. So follow along and be honest with yourself if you miss them. We're going to call this the practice round. So whatever grade you get on this one, it's not real. I just want you to see what you would have got. So please be honest with yourself. Then I'll give you a second set. It'll be the real deal that we'll actually grade closely on Monday. 4 solutes, oxygen and carbon dioxide, can pass through the membrane. Sugar and salt cannot. All right. So for beaker number 1, there's no membrane. Same with number 2. What happens? What goes where? Come on. It's like the candle on the table. It goes outward from where it's at, right? So your arrows should go out from the center. And you should say the salt in this case and the oxygen in this case move outward. That would be the right answer there. Number 3, oxygen outside but not inside the cell. What goes where? Chris. This one. What goes in? Make sure you specify in your answer what is going where, because remember, sometimes it's water. So oxygen goes in is the correct answer there. Number 4, bunch of salt outside the membrane. Diane. Yeah. The water will, you say, leave the membrane? Exactly. Water goes out. Very good. Because the salt wants to go in, but it can't, right? So now we have to think about water. You can either think about it going from high to low water concentration. Well, middle has no salt, so that's lots of water. going to less water. Or you can think about it, the salt wants to go in, but it can't, so the water goes the opposite way. Good, good so far. Number 5, we've got carbon dioxide inside. Jane, what goes where? It's like the membrane's not even there because carbon dioxide can cross. So the carbon dioxide diffuses out of the cell, CO2 out. 6, whether I drew it well or not, it's supposed to be the salt being basically equally concentrated inside and outside the cell. So and this is a tricky one. This is one that you have to kind of word correctly. What would you say, Benny?
I was confused on this one.
Yep, because we haven't really talked about what you say when, like, some of you probably said nothing happens.
Yes,
And that's
what I said.
You got the right idea, but it's never that nothing happens. What's really happening is random motion is still happening, but what's happening here is water is going in and out equally. That's the best way to say it. Water goes in and out equally. Riley, what did you say?
Well, I was confused on that one too, but I was kind of what I was thinking.
So that one basically is already at equilibrium, and so water is moving in and out equally. Number 7, we threw you a curveball, and there are 2 different solutes. What did you say? True? Exactly. That's perfect. What you got to do in these is basically look at the one solute like the other one's not there. So you did perfect. You said, okay, the CO2, there's more of it out here than here. It can cross. CO2 goes in is correct. But also, there's more salt outside. There's none inside. We also have to think about water. So aside from CO2 going in, if we look at the salt like it's not there, but water goes out. CO2 in, water out are the 2 correct answers for that one. And finally, number 8, Riley, what'd you have? Water goes in because there's more water here and less water here. Even though there's some sugar, this is higher concentration of water, this is low. So out and water goes in. Excellent. Next side, the horizontal side, sorry. So this one here with the oxygen inside. Reagan.
The oxygen will go out.
Oxygen leaves the cell, perfect. Next one with the sugar outside. Nathan. This one here.
It just stays on the outside.
The sugar does stay on the outside, but that sets up the osmosis thing where water moves. Which way does the water go? Water goes. What's left? What's left? Not in. It's water goes out. Yeah, yeah. Because remember, it goes the opposite way the sugar wanted to go. So the sugar wants to go in, but it can't. So the water goes the opposite way. That's how you do that. Salt inside the membrane.
The water, I can't decide.
Water goes in. Perfect. Moving right along. We've got another 2 for here with sugar inside and then oxygen kind of spread equally. McKinley? Well, it's oxygen, but sugar can't leave. Remember, sugar's stuck. So now we have to think of water. So, sugar can't leave. It's stuck. So we have to think about water. Which way does water go? Yes, water goes the opposite way the sugar wanted to go. So water goes in, and then I know it might not look like it, but I tried to draw the oxygen equally concentrated. So that second answer, water goes in is the first answer. The second answer is oxygen goes in and out equally because it's already at equilibrium. Okay? This one here. Annalise,
I don't know.
you don't know?
No.
That's cool. Come on. Water goes in and out equally because there's no difference in concentration here. So water's always moving. So water goes in and out equally here. And then hopefully you didn't let numbers intimidate you here. All you got to know is high and low, Riley.
Would the water goes in because there's
So you're right that water moves. It's 60% sugar outside.
Water goes out because there's more water in the cell.
Right, 10% sugar means 90% water, right?
Yeah.
So more water here than here. Water goes from in to out. Water leaves the cell. And for the last one, 50% oxygen outside, 80% inside. What goes where, Tristan? Water would go inside, except that it's nothing stuck here. Oxygen can cross the membrane. We don't have to worry about water at all. So what goes where? Oxygen goes out from 80 to 50, high to low concentration. Good. Oxygen goes out. Okay, score yourself. It'll be out of, what, 7.5, basically, because there were 15 of them. We'll take a half point for anything you missed. I'd just like to see the totals. Cool. So you are going to get a second set of beaker problems here momentarily. That will be the real grade. It'll be a little more complex, but not much. You'll be fine if you kind of take in mind what you missed here. You'll also get sample essay questions tonight, even though we haven't finished the last bit. That'll be one of them. The rest of them will be fine. We are looking at those being due on Tuesday and your test being Wednesday. Any questions about that? Well, I guess you better go get your eggs then. We better do our real live beaker problem. And go ahead and open up Classroom and find your egg lab sheet in there. Yes, sir. The egg?
Yeah.
No, the vinegar, and I know this because somebody dared me to do it last year. And I did it. I went ahead and cooked a shellless egg that had been soaked in vinegar. Hey, maybe it'll taste like a pickled egg. Maybe it'll be good. It was not good. If you want to do it, I recommend maybe adding some sugar to the egg or to the vinegar, and maybe some salt as well. That's more of a pickling situation. Just vinegar, it just made it taste like a vinegary egg. It was gross.
Something more than that. I was like, in my backyard, he just
Yes. Now the urge to throw these at something is strong. I know it. I have it, but we're going to have to not do that. Remote learners, I can't control what you do. Tell me stories if you're doing these fun. I've always said that, like, if I know I'm gonna retire, quit teaching, like some year, I'll just have all my students grab their eggs and throw on something all at once, and I think that would be very satisfying. But we're not there yet. Okay, open that thing up.
G.
We should see. G. G.
There's like G. Just think it's
Okay. Oh, did it like auto number letter?
just random G.
Hey, geniuses, G stands for grams.
Oh, I bought it. I did that on purpose. I think. I went for experience. I had this.
Okay, so for egg preparation procedures, first of all, share it with your partner. So one of you share with the other one, so you're working the same document.
Oh my goodness. See, if I was those,
That's right. These kids give us
exactly.
I just like how all the answers
It's so funny.
Okay, once you've shared egg preparation procedures, you've done that. For egg preparation procedures, please type in put egg in plastic cup, cover egg in white vinegar for 24 hours.
Would it be different from other types of vinegar?
Yeah, because there's sugar in some types of vinegar, so that might change things a little bit. And I haven't done it, so that's something you can do at home. So, cover egg in white vinegar for 24 hours in a plastic cup. That's all you got to do for that. Now, your procedures for today are a little more complicated than yesterday. Because you are going to, the way we're going to determine whether osmosis really happens is we are going to weigh these eggs before and after we put them in a solution. I will tell you right now that solution 1 is corn syrup.
Yeah.
Corn syrup, which is basically what you get when you use an industrial facility to blow apart field corn, which is the stuff most cornfields have that's not good to eat. What they do is they break it down into its components, and the sugar that they get out of this is corn syrup. This is what you have on your pancakes, by the way, unless you're spending, $30 a bottle for real maple syrup, the runny stuff that really tastes like maple. What you have for your syrup, Mrs. Butterworth's or whatever, is this with brown food coloring and some artificial maple flavor. Some people just put this on their pancakes. It's just good. Anyway, this is a concentrated sugar solution. You are immersing your egg in a concentrated sugar solution. That makes this a giant beaker problem, doesn't it? You should know what's going to happen to something with a membrane when it's put in a concentrated sugar solution. That's the hypothesis part, right? The mass part is you are going to do this. You're going to take, one of you is going to take your egg to the sink. You are going to put your fingers over top of the cup. You are going to dump the vinegar into the sink and dump the egg into your hand. The egg will still have some shell. Shh, listen. The egg will still have some shell on it. Don't worry about getting that off. What you want to do is run the sink and get a gentle stream of water going and rinse your egg and rinse your cup off. You don't have to worry about rubbing the shell off. That's a quick trip to popping your egg, and we don't want that, right? You know about our sinks, right? They're booby traps. So your egg mass before immersion in solution 1 in appearance will come next. Then you do your hypothesis. Then you're good. You put your egg, you bring your egg to me, and I cover it in corn syrup, and you put it back where it goes. Any questions? All right.
So for the hypothesis, do you want us to do that first?
You can do it whenever you want, just before tomorrow. Yeah. Go.